All right, so today we're going to dive into something I know you've been curious about. It's how cannabinoids interact with those TRP channels, especially TRPV1 and TRPM8.
Right.
Sounds like you're ready to move beyond just CB1 and CB2 receptors, and you're definitely no stranger to the complexities of cannabinoid signaling.
Mm.
I mean, the research you sent over, this is PhD level stuff. We're talking GPCR activation, subunit dissociation, CMP, PKA. It's a whole alphabet soup of molecular players. But don't worry, we're going to break it all down together.
Absolutely. It is a complex landscape for sure, but it's also full of some really exciting insights.
Exactly. And you mentioned wanting to kind of explore the history of cannabinoid research a bit. I think it's worth starting with the groundwork laid by Dr. Rafael Mechoulam. His work is pretty foundational to the field. Back in the 60s, he and his team synthesized A6-Rion tetrahydrocannabinols. And then in the early 90s, they were really instrumental in identifying anandamide and 2-AG, which are those endogenous ligands for the CB1 and CB2 receptors. That really kind of opened up the floodgates for understanding the endocannabinoid system.
Absolutely. Mechoulam's work was a huge turning point. It sparked a wave of research into the endocannabinoid system and its role in everything from pain and inflammation to mood and appetite. But what's really fascinating is that the story doesn't just end with CB1 and CB2.
Right, that's what's really interesting for you, right? The research you sent over suggests that some cannabinoids might be doing their thing even without those familiar CB1 and CB2 receptors. It's like there's this whole other hidden world of interactions going on.
Yeah, it seems that way. And this is where we get into the concept of non-classical cannabinoid receptors. Essentially, these are receptors or pathways that cannabinoids might be activating independently of CB1 and CB2. For instance, some studies have shown that certain cannabinoids have antioxidant properties, and that might contribute to their therapeutic benefits.
So we've got these cannabinoids potentially acting on additional receptors, like GPR55, for example. What are the implications of that? Why does this even matter?
Well, think about it this way. If we have more targets for cannabinoids to act on, that opens up a whole new world of possibilities for developing more targeted therapies. It's like we're expanding our toolbox for treating a wider range of conditions, potentially with fewer side effects.
Okay, so let's get a little deeper into how these receptors actually work. We're talking about GPCRs, those G protein-coupled receptors. The research you sent over gets pretty deep into the complexity of them, even mentioning how a single amino acid change can completely alter how a receptor binds to its ligand, and ultimately what happens inside the cell.
Yeah, it's true. GPCRs are incredibly intricate, and these tiny changes can have a huge impact on the downstream effects of activation.
So how do these GPCRs work? Can you give us kind of a simplified overview of this complex process?
Sure. Imagine a chain reaction. When a cannabinoid, or any ligand for that matter, binds to a GPCR, it triggers a conformational change in the receptor. This sets off a cascade of events involving G proteins, which then interact with other signaling molecules within the cell. Think of it like a domino effect, with each step leading to the next, ultimately influencing a particular cellular process.
Okay, that makes sense. But the research also mentioned something called functional selectivity. What does that mean?
Oh, that's a really important point. It highlights the fact that even if two different agonists activate the same receptor,
Right.
they might not necessarily trigger the same downstream effects. They could actually lead to different cellular responses. It's like hitting different notes on a piano. Even though you're using the same instrument, you can create a wide variety of sounds.
So how does that play out in the real world? Can you give us like a concrete example?
Absolutely. Think about morphine and Damju. Both of these substances activate the mu-opioid receptor, or MOPR, which is involved in pain relief. But they trigger different desensitization mechanisms. Damgio primarily relies on a pathway involving GRK and arrestin proteins, while morphine tends to favor a pathway involving PKC or protein kinase C. These subtle differences in how they activate the receptor can influence their effectiveness and potential side effects.
Wow, that's pretty fascinating. So even though they activate the same receptor,
Yeah.
they can have different effects on the cell.
Exactly. And understanding this concept of functional selectivity is really important for drug development. It means we can potentially design drugs that target specific pathways or cellular responses, leading to more targeted treatments and hopefully fewer side effects.
So it's not just about finding a drug that binds to a receptor. It's about understanding the nuances of how it activates that receptor and the specific effects it triggers.
Right, exactly.
Okay, so we've laid some of the groundwork for understanding these non-classical pathways. Now let's bring it back to the main players you were interested in, TRPV1 and TRPM8. These are the TRP channels that really caught your attention, right?
Yes, exactly. Both of these channels play crucial roles in how we perceive the world around us, particularly when it comes to temperature and pain.
So tell us more about these channels. What makes them so special?
Well, TRPV1, for example, it's activated by heat and capsaicin, that fiery compound that gives chili peppers their kick.
Ah, so that's what makes us feel that burning sensation when we eat something spicy.
Precisely. But it's not just about spicy food. TRPV1 is also involved in inflammatory pain signaling, which makes it a really important target for pain management therapies.
And what about TRPM8? What does that channel do?
TRPM8, on the other hand, is activated by cold temperatures and menthol. It's what gives us that refreshing, cooling sensation. Think about that blast of fresh air on a chilly day or the minty feeling from chewing gum.
Okay, so TRPV1 is all about the heat and spice, while TRPM8 is about coolness and refreshment. But how do cannabinoids fit into all of this?
Well, the research you provided suggests that cannabinoids can actually modulate the activity of both TRPV1 and TRPM8, and that's where things start to get really interesting. There's evidence that certain cannabinoids, like cannabidiol or CBD, can interact with both of these channels.
So we're talking about the potential for CBD to influence both our perception of temperature and pain.
Exactly. And the way it interacts with each channel seems to be quite different. Some studies suggest that CBD might act as an antagonist at TRPV1, meaning it could potentially block its activation. This could have implications for reducing pain and inflammation.
So it might actually dampen that burning sensation. That's pretty amazing if you think about it, potentially using CBD to target specific pain pathways.
It is promising, and researchers are actively exploring its potential for treating conditions like arthritis or neuropathic pain. But the interaction with TRPM8 seems to be a bit different. Some studies suggest that CBD might actually enhance TRPM8 activity.
So instead of blocking it, it's actually boosting that cooling sensation. What are the implications of that?
Well, it could potentially have therapeutic benefits for certain types of pain that are sensitive to cold. It's an area that requires further research, but the initial findings are definitely intriguing.
It sounds like we're just starting to uncover the complexity of these interactions. It makes you wonder what else is going on with other cannabinoids and these non-classical pathways. Does the research shed any light on that?
We're still in the early stages of understanding the full scope of these interactions, but the research does point to the possibility that other cannabinoids might also be modulating TRP channel activity. For example, there have been studies looking at the effects of THC, the psychoactive component of cannabis, on TRPV1.
So even THC might be playing a role in these sensory pathways.
There's some evidence of interaction, yes, but more research is needed to understand the precise mechanisms involved and the potential therapeutic implications. It's a complex puzzle, but each piece we find brings us closer to the bigger picture.
It's amazing to think about all these interconnected pathways and how they all contribute to our overall health and well-being. It really highlights just how intricate the endocannabinoid system is.
Absolutely. And the complexity is both a challenge and an opportunity. It means we have a lot more to learn. But it also suggests that there's enormous potential for developing new and innovative therapies for a wide range of conditions.
Okay, so let's pause here for a moment and try to synthesize what we've learned so far. We started with the idea that while CB1 and CB2 receptors are key players in the endocannabinoid system, they're not the whole story.
Right. There's growing evidence that cannabinoids can influence a variety of other receptors and pathways, including these TRP channels like TRPV1 and TRPM8, which are so crucial for how we experience temperature and pain.
And what's really exciting is that these non-classical pathways could hold the key to developing even more targeted therapies for pain, inflammation, and potentially even a whole host of other conditions. It seems like we're on the cusp of a new era of understanding when it comes to the therapeutic potential of cannabinoids. We've talked a lot about the potential benefits of targeting these non-classical pathways, but what about the potential downsides? I mean, are there any risks or challenges we should be aware of?
Well, it's kind of like navigating uncharted territory. With any new therapeutic approach, there's always a chance of unforeseen side effects. And because these pathways are so complex and interconnected, we need to be cautious and do our homework. Thorough research is crucial to fully grasp the potential risks and benefits before we can even think about widespread applications.
Yeah, it's a good reminder that we're still at the tip of the iceberg. When it comes to understanding the full scope of the endocannabinoid system, there's so much more to discover.
Absolutely. That's what makes this field so exciting. Every new discovery opens up new avenues for exploration and innovation. It's like peeling back the layers of an onion. There's always something new to uncover.
Okay, so thinking about all of this makes me wonder, if the endocannabinoid system is involved in such a wide range of processes, could targeting these non-classical pathways lead to more effective treatments for conditions beyond just pain and inflammation? Could it hold the key to treating things like anxiety or depression, or even cancer?
Those are all fantastic questions, and they're questions that researchers are actively investigating. For example, some studies suggest that GPR55, one of the non-classical cannabinoid receptors we talked about earlier, might be involved in cancer cell proliferation and metastasis. So targeting this receptor could potentially offer a new approach to cancer treatment.
Wow, that's pretty mind-blowing.
Yeah.
It really seems like the possibilities are endless.
They really are. I think it's safe to say that the next decade of cannabinoid research will be filled with groundbreaking discoveries and innovative therapeutic applications. It's a really exciting time to be following this field.
I'm definitely eager to see what the future holds. And a huge thank you to you for guiding us through this complex and fascinating topic. This deep dive has been incredibly insightful.
The pleasure was all mine. And to our listener, I really encourage you to continue exploring this incredible field. The more we learn about the endocannabinoid system and its intricate pathways, the closer we get to unlocking its full therapeutic potential.
That's a wrap for this deep dive. Until next time, keep those brains buzzing and stay curious.